Hi everyone, welcome back to my channel. I am Mahnur. Today, I'll take you inside the lab to show how we perform in siRNA transfection, a powerful method used to silence or knock down a specific gene inside cultured cells. So, what exactly is siRNA? siRNA stands for small interfering RNA, a short double-stranded RNA molecule that can specifically target and degrade messenger RNA, or mRNA, preventing the cell from producing a certain protein. This technique is extremely important in gene function studies, disease modeling, and even drug discovery, because it helps researchers understand what happens when a gene stops working. In this video, I'll demonstrate how to perform siRNA transfection using Lipofectamine, a reagent that helps siRNA enter the cell without damaging it. We'll be using U2OS cells, a human osteosarcoma cell line, and 3 siRNAs, a control siGFP, siRSF3, and siSIAF2 to study gene knockdown efficiency. So let's get started step by step. Materials required for siRNA transfection.siRNA, control siRNA, 2 target genes siRNA, transfection reagent, e.g. Lipofectamine RNA-IMAX or Lipofectamine 2000, U2OS cells, Opti-MEM, reduced serum medium, 6 well tissue culture plates, sterile complete media, short phosphate buffered saline, PBS, trypsin EDTA, microcentrifuge tubes, 1.5 ml, and biosafety cabinet. On the first day, you have to seed 30%. U2OS cells in 6 well plate for siRNA. Transfection experiment that we will do on the second day. 1. Put complete media, PBS, and trypsin EDTA into hot water incubator at 37°C for 30 minutes. First, always clean your workspace using 70% ethanol or your lab's disinfectant. Make every arrangement on biosafety cabinet. Take out your cells from CO2 incubator and check the confluency if the cells. Confluency is around 70 to 80% it's ready. For cell transfection. 2. Remove the old media from the cell plate and add PBS 2 time for washing. Three, add trypsin EDTA 2 ml for detaching cells and put it in incubator for 2 to 5 minutes for complete detachment. Label each well clearly. Control siSRSF3, siSIAF2, and co-transfection. Better to seed your U2OS cells into a 6 well plate around 30% so they reach 60–70% confluency on the day of transfection. This confluency ensures that the cells are actively dividing but not overcrowded, which gives higher transfection efficiency. Add 2 ml media in each well and count cells in Hemocytometer to know how much is the total concentration of cells and how much cells you need to add in media. 4. After 2 to 5 minutes, take out your cells from incubator and check the detachment. In microscope. Neutralize trypsin with 4 ml complete media and mix it properly. After neutralization, transfer all the cells into centrifugation and discard supernatant and mix 1 ml media and do cell counting. Then transfer cells up to 30% to each well. Check the cells in microscope and put it in CO2 incubator for tomorrow experiment. next day. Put all the required materials such as Opti-MEM, siRNAs, and Lipofectamine RNAi Max in biosafety cabinet. Take 4 microcentrifuge tubes and label them as SiGFP, SiSRSF3, SiSIAF2, and co-transfection. Add 200 UL of Opti-MEM to each tube. Then pipette 2 UL of 20 siRNA into each corresponding tube. For the co-transfection sample, add 2 UL of SiSRSF3 plus 2 UL of SiSIAF2, making a total of 4 UL. Mix gently by pipetting up and down or by a brief vortex, then incubate these siRNA dilutions for 5 minutes at room temperature to stabilize before adding lipofectamine. Now, prepare your lipofectamine reagent for complex formation. In a new set of tubes, add 4 UL of lipofectamine into 200 UL of Opti-MEM for each condition. Gently mix the tubes by hand. Avoid strong vortexing because it can break the lipid complexes. Let it rest for 5 minutes at room temperature. Then slowly add the diluted lipofectamine to the siRNA mixture tubes. Mix gently and incubate the complexes for another 10-15 minutes to allow them to fully form. Finally, add the siRNA lipofectamine complexes to the cells. Gently pipette 200 UL of each complex solution into the respective wells of your 6-well plate. Swirl the plate gently for even distribution and return it to the CO2 incubator at 37 degrees Celsius. Record your plate map and experimental details in your lab notebook. After transfection, monitor your cells under the microscope. You can collect samples after 24 hours for RNA or 48 hours for protein to check knockdown efficiency using qPCR or Western blot, and that's the complete siRNA transfection protocol. If you found this video helpful, don't forget to like, subscribe, and comment below. Stay tuned for more lab tutorials and behind the scenes from my research in Korea. Thank you!